So, hello world. My name is Miles, and this is a bi-weekly or maybe daily stream. I'm not quite sure yet, where I browse the latest scientific discoveries on the websites for top scientific journals. And if I have any viewership, I try to let the viewers pick what kinds of papers we talk about or what kind of research that we want to look at. Today, though, at least for now, I'm completely and utterly alone, so I'm just going to look at whatever papers I want to look at. I'm trying to set up, see, I'm trying to set up chat right now on my phone so that I can keep a lookout in case anyone joins. I actually had a guy watch my last stream. Like the first stream I ever did, I actually had a viewer. I was so surprised that uh, that anyone wanted to come here. Uh, Not that um, I was just blown away really because I've never done this before, as you can probably tell pretty obviously, and um, the video quality I know on the stream is very subpar, but uh, here goes nothing. So I'm just gonna go browse subject section, and we'll see what comes up. Actually, for people that drop in on the chat, try and send them a message, um, just so that they know what the stream is actually about. I only have one viewer right now, and I'm pretty sure that's me. So let's just go ahead and do that real quick before I get into actual science. Can I just say figures? Okay, almost done. Okay, there we go. That way, people join, they'll actually know what's up. They won't just, you know, see a random guy talking about who knows what and decide to leave. So, okay, back to what we were doing. So, I'm a biologist by trade, although I'm also a self proclaimed mathematician and chemist, but most of my interests lie in biology, so a lot of the papers I read are probably going to skew towards biology. I especially love genetics, so we're going to start our day by looking at genetics papers. Okay, so I'm just going to browse through these and uh, find a title that kind of pops out to me, and then look at the figures associated with that paper and try to figure out what the research is just by the figures. So let's see. Oh, this one kind of pops out to me immediately. Functional insights into the fractal structures of the heart. Okay, that sounds interesting. I mean, I don't know much anatomy at all, but I know what fractals are, so maybe we can try and piece together what's happening here. Find the figures. Bring full-size images. One thing I love about nature is that their figures are always top-notch. Like, I think I've said this before, but on a previous stream, but uh, scientists are really great at making graphs, especially ones that not just ones that communicate data well, but also graphs that are just aesthetically pleasing. And so let's try to figure out what's going on here. Here we have what's probably a cross section of a heart. This arrow here is pointing at myocardial trabeculae. Oh man, yeah, I obviously have very little knowledge of anatomy, but yeah, that's definitely it's definitely some flesh. So here got more cross sections of a heart, so kind of just that, but in cartoon form, I guess. Let's see, we have kind of pixelated versions of ventricles, which I think are heart valves, if I remember correctly, from my intro biology classes. And now we go from heart valves to counting boxes over here on the right. So it looks like they're tracing the outline of the ventricles by dividing up the outline into these box regions, which is interesting. Wonder why they do that. And then, let's see here, or I guess this kind of gets at the fractal part of the research. So I think fractals, let's look it up real quick. Fractals. I'm just going to go to the Wikipedia page so we have a nice picture to reference. Okay, so this is probably one of the more famous fractals. Set, I think you call it. And it's essentially, just based on what I know what fractal is, I'm definitely not an expert, but a fractal is like a pattern that repeats forever, no matter how far you zoom in. At least that's kind of my understanding. So if you were to zoom in on just a portion of this Mandelbrot set here, then you would see similar patterns, I think, to what we see in this full picture. But you'd see patterns like that no matter how far you zoomed in. So yeah, it looks like there could be some kind of similar structures going on in the heart. So here we have, so I'm back on the figures for the paper and comparing some aspect of the fractals between males and females. It's like this peak here for males is a little larger than it is for females. Not quite sure what the height of the peak is supposed to represent. I guess it's frequency of some kind. So now fractal dimension again, but now it's varying with age, with height, with weight, and with body mass index. Fractal dimension seems to be maybe slightly increasing with height and weight and BMI, but not age really. So body mass index is a measure of weight really, except it's normalized by a person's height. So you can see here that BMI is measured in units of kilograms per meter squared. And so kind of what the usefulness of a BMI measure in something like this, from my understanding, is that it helps show associations between a person's weight and another variable while controlling for someone's height, since height and weight are very often correlated. People that are taller also tend to be heavier. See, along the tops of these little plots, we have some kind of hump. I'm guessing that's showing the density of data around a particular value on the horizontal axis. So like in this BMI plot here, we have dense region of points around here in the plot, so we see a pretty tall peak there. Okay, that's interesting. Let's look at the next figure. Go to figures. Right, figure 2, genetics associations. All right, this looks cool. Now, so these plots are pretty common in genetics literature. This panel A over here is what's called a Manhattan plot. And what it shows associations between particular regions in someone's DNA, which is shown here on the x-axis, and some trait that scientists are measuring. And so points that fall above this horizontal line here show those indicate regions in someone's DNA that have very strong associations with a particular trait. So points that fall above these lines tend to map to genes or some other regions of the genome that regulate whatever trait is being investigated. So it looks like the authors identified a number of genomic regions that correspond to the fractal structure of the heart. However, they measured that, which I guess was showed in the previous figure. Their fractal dimension, I guess, was the trait that they're investigating. Each of these strings of text here is probably a different. Each of these strings is probably a different name for a different gene. So TNNT2 is probably a gene, as well as TTN, and so on. This plot here looks very similar. Again, we have, except now we're focusing on just one chromosome. Here, all DNA from all of the human chromosomes were included on this x-axis, with the different chromosomes corresponding to alternating gray and light gray and dark gray colors. For some reason there's like a valley here for chromosome 9, which is interesting. It's like this part of chromosome 9 is just missing or something. But anyway, I guess that's not unheard of. From my understanding, the human genome isn't complete, so it could certainly be missing regions here that weren't included in the study. But over here, we have something pretty similar, except now we're just focusing on one chromosome. So these dots mean the same thing still. Each is a different region of DNA that they tested for association with some trait. So here, the higher up a particular point is, the stronger the evidence is that that genomic region plays or is associated with the trait being investigated. So they identified a number of important traits here. With here they're showing co a, a statistical quantity known as the coefficient of variation, I think, or maybe coefficient of determination. R squared is commonly how it's denoted, although normally you'd see a capital R, so that might be something different. Either way, seems to be showing um, points with these lighter colors have stronger associations with this fractal structure trait or fractal dimension. And then here, oh man, these are getting deeper, so not entirely sure what's going on here. We have slice and meta on our two axes. Let's see if there's anyone in the chat. No, no one. Like, I think I mentioned this earlier, but my last stream, actually, one guy showed up and he was talking to me in the chat. Sadly, I wasn't paying attention to the chat at the time, so he just left. But hopefully, someone like that will pop in eventually. If not, though, I'm totally fine just talking to myself and looking at papers. I already do that every day anyway. Now I just record it and post it on the internet for everyone to enjoy. And then finally, let's see here, and box and whisker plots. And I rarely ever see box and whisker plots, but here they are. So these are showing like distribution of fractal dimension along along these slices here, I think. Okay, so slice, it could be like cross sections of the heart, I'm guessing. And then they measured fractal dimension at a bunch of different heart cross sections. And here they're showing their distributions. So you can see that fractal dimension, or really variance in fractal dimension as well as mean, varies along cross sections of the heart. Don't know in what dimension along the heart they took their cross sections. I assume it was like kind of like what we saw in the first figure here, top to bottom. Oh, and here they're mapping out their basal, mid, and apical regions, which we kind of saw in that previous figure. Okay. Next is knockouts and cardiac stuff. Let's see here. I'm not sure what this is. Cardiac trabeculation. Let's go ahead and let's Wikipedia that. Some medical terminology. Let's see here. Trabecula. It's a small microscopic tissue element in the form of a small beam that supports or anchors a framework or part of a body. Okay. So it's like it's analogous to a structural beam. So here so in this figure here, the authors knocked out some gene, and that led to a reduction in these cardiac support beams in something called Medaka. That's probably another anatomy term. Let's look it up here. No, actually, first result I got was a Japanese rice fish. I mean, I don't think the authors are studying rice fish. I mean, maybe they are right here, actually. I know that fish are pretty common model organisms for certain types of human genetics, which is surprising considering that fish and humans don't seem terribly closely related, but it's true, especially zebrafish. Zebrafish are pretty common. Yeah, Medaka seems to be a Japanese rice fish, which I was really not expecting. I was thinking that it was some kind of heart structure. Um, so let's try to figure this out here. So we have fraction and then different genes. So I guess these are fractions. Okay, so these are fractions of their study population that have either a normal phenotype, which is a phenotype is the same thing as a trait, um, a phenotypic phenotype. I'm not quite sure what that means. It seems pretty redundant. And then, or they're just dead. So looks like knocking out. Okay, so it looks like knocking out MTSS1 killed quite a lot of the fish. And then, but fish that, let's see, M. cherry. M. cherry, that's some kind of like dye or something, I think. Not entirely sure. Some kind of fluorescence, a fluorescent molecule, M. cherry, I think. Okay, and this next figure, I'm just gonna skip over that one. This next one, have these same mutants again, and now we have different phenotypes on the y-axis, different mutants on the x-axis, and then sample size as well, right above the x-axis. Looks like we have different point sizes for how many individuals fill into each category. So knocking out MTSS1 was sublethal for many individuals, but also resulted in abnormal heart morphology and a reverse looped heart, which sounds kind of crazy. Silent heart, oh man. Knocking out TNNT2A resulted in a silent heart? Man, what does that mean? Silent heart. Sound, whatever it is, it does not sound desirable. So let's see here. CRISPR knockout. So you don't know what CRISPR is. You may have had your head under a rock for the past few years, but it's a very common, also somewhat controversial, at least in the realms of human genetics, gene editing tool. Allows scientists to alter pretty much any region of DNA that they want, as long as they know its sequence, or in other words, as long as they know its structure. Different embryos, comparing them, and then I'm not entirely sure what this is. I assume it's some kind of heart cross section or some kind of scan of a heart. So it's showing the inside, showing the outside. This is what a normal heart looks like, and this is what happens when you disable this MTSS1 gene. See that I guess the heart flow is altered. You don't see as much of those supporting structures in this heart as this one, which kind of goes back to that last figure we saw. Okay. Next one. I don't know if I'll go through all of these. We'll do one more. Yeah, because there are a lot of figures, especially in nature papers. So we'll just do one more, and then we'll go on to the next one. See what I'm doing for time. Okay, like 20 minutes. So, you know what? We're just going to go on to the next paper because I've been looking at this one for a while. Okay, geez, I'm just clicking on everything. Let's go back to the browse subjects. So I listened to the previous stream that I did, And man, I'm very sorry for whoever has to watch that. I guess it's not that bad, but my voice is super quiet. Uh, Not entirely sure why. I mean, you can still hear it. It's just it's just very quiet. I have to mess around with the settings there. Yeah, it's taking forever to load. Okay, here we go. Um, Back to genetics. Let's see here. Oh man, Tuatara genome. Okay, we gotta check that one out. I only vaguely know what a Tuatara is, but it's a pretty weird organism. Let's get a picture of one real quick. Yes, it's one of these it's a reptile. I've heard of Tuataras before. I can't remember where. I think it was in some YouTube video. Man, he's adorable. Oh, look at that little face. Looks so happy. No. Anyway, scientists sequenced the genome of one of these fellows. Reveals ancient features of amniote evolution. Let's see what an amniote is real quick. Clade of tetrapods. Okay, so an amniote is an animal that lays eggs on land. Okay, I should have known that. I probably learned that at some point. Anyway, let's look at the figures. Oh, so here they already have a picture of a tuatara right here. So I guess this is probably showing distribution of tuataras. I thought these guys were in New Zealand or something. Maybe that shows where captive tuatara are. I guess this is where they got the genome on Lady Alice Island. Um, so this is pretty cool. This is a phylogeny showing relatedness between different sets of organisms according to evolutionary theory. And I guess this is tuatara right here, this little yellow guy. So tetrapods are split off into amniotes and I guess organisms that don't lay their eggs on land. Surprised, I would have expected it to be a little less closely related to mammals. Well, it is pretty well diverged. That's pretty cool. It seems to be entirely different from what the rest of these reptiles. Actually, I think I think that's what's special about Tuatara. I think they're like the last member of their genus or something. Um, and they're just very unique. There's um, nothing else alive that's closely related to them. So anyway, that's pretty cool. Let's try the next figure. So here, we're talking about the repeat landscape in the Tuatara genome, evidence of recent activity and a greater expansion of diversity of repeats than any other amniote. So here, they're talking about the repeat landscape of the genome. What that means is that when you look at the structure of DNA in a living organism, structure is very often composed of repeating patterns. Um, so if you have, especially in regions that don't encode for any kind of trait, so it's pretty common to look at a region of the genome and see just like a single structure that repeats many times. And really, these repeats, they don't really code for anything, as far as I know, but they are very abundant in most eukaryotic genomes, most genomes of organisms that aren't bacteria. And so looks like they may have constructed another kind of phylogeny where they are using just those repeats. It's pretty interesting. And this is what's called an unrooted phylogeny. So we saw in that previous figure the phylogeny had like a distinct tree-like structure to it, whereas this is more of like a sprawling web. The first kind of phylogeny is called a rooted phylogeny, and this one's called an unrooted phylogeny. Because in these unrooted phylogenies, you're not how do I put this? Don't typically have, not showing like the common ancestor of all of the members in the phylogeny when you use an unrooted phylogeny. So let's see here. Now they're comparing different DNA sequences, I'm guessing, in this sign family. I'm guessing the sign is a family of repeats, perhaps. They're showing how different each of these repeats are across these different families. It's just a guess. And then here, several different clades of repeats, perhaps. Clade is just a group of closely related organisms. I'm not sure if it has a more rigorous definition than that, but let's see here. Now we're getting to sex differences between Tuatara. Here we have comparing males and females and methylation here on the y-axis. Now, so explain what that is. So in DNA, so if you've had some kind of intro biology course, you'll know that DNA is really it's a molecule that's made up of 4 different components, or really building blocks. You can think of them as Legos almost. And those building blocks come in 4 different flavors, denoted as A, T, G, and C. And these different, so DNA is just a long string of those 4 different building blocks. But those building blocks can be slightly modified. So here they're talking about C-G methylation. So C and G are 2 different building blocks of DNA, and methylation is a kind of modification. And what methylation typically does is it prevents genes from being expressed by a cell. So here, showing levels of methylation between males and females, and it seems pretty similar between them, although it looks like females have slightly more variation in how much methylation is in their genome, which is pretty cool. And now, okay, so this is another Manhattan plot. I feel like I've looked at 1,000 of these today, and Again, on the x-axis, we have different different regions of DNA, and on the y-axis, have the strength of evidence for them being associated with a particular trait. I don't know what trait they are measuring here in this instance, but um, something. It looks like none of these DNA regions are significantly associated with a trait. Effective population size. So we saw this in like the lion paper. If you watch that stream, it's posted on YouTube, by the way, if you ever wanted to watch it. But yeah, it showed a very similar plot for lions, where um, effective population size of lions has just plummeted over time. Looks like showing something similar here for Tuatara, had this huge dip about 1,000,000 years ago, and recently rose back up, have another dip again. And finally here, we have what's called a principal component plot. And that just, principal components are, kind of think of them, or I tend to think of them as kind of representations of many variables that have been collapsed down into a single variable. So it's a method of making your data set easier to work with. And now, instead of who knows how many variables the authors were looking at, they just have 2. Whereas before they may have had like, I don't know, 20 or 50. Now, to visualize their data, they just look at these 2 variables, which capture most of the variation in their data set. And finally, I'm not sure what this is. This little red, green, and blue diagram on the right. On this PCA plot, you can see how Tuatara from different regions of these islands differ from one another. Like Tuatara on the North Brother Island are all very similar to one another, which you can tell by all these points being clustered together. But Tuatara on Stevens Island are very similar to other Tuatara on the Stevens Island, but very different from Tuatara on the other 2 islands. Let's see here, how am I doing for time? Okay. Maybe look at one more figure. Oh man, I'm tired. Ugh, I really need to get back on my normal sleep schedule. Everything to do with this pandemic, working from home all the time, is really throwing everything out of whack. Let's see here. I'm just getting distracted by the figures. I actually load it. There we go. Okay, this looks cool. GOC score distribution. Species. Okay. Okay, so GOC stands for gene order conservation. So how well the order of genes in an organism's genome is conserved across many species, I guess. Perhaps higher numbers of GOC score refer to larger amounts of conservation. Let's see, where's the Tuatara here? I think the Tuatara are in yellow. And oh man, they have alligators on here. Oh man. Ugh. Not quite sure about that. Let's see here. Last common ancestor of Tuatara. Percentage of genes in a conserved position. Gene order conservation, pairwise comparisons. So it looks like how well gene order is conserved varies a little bit with what ancestor we're looking at for the Tuatara, which is interesting. Hmm. All right. Well, I think I'm gonna take a nap. Oh man, I'm tired. But if you're watching this at a later date, whoever you are, thanks for sticking around. And I'll be back. I'm not sure. Maybe tomorrow, or I want to do a bi-weekly schedule. So every every Saturday and Sunday at 11 a.m. Eastern time, I think would be a good schedule. But um, for now, since I have holiday coming up, might might stream tomorrow as well. Yeah, I don't know. I'm still figuring it all out as I go, but I'm hoping to make this a regular thing, at least twice a week. Anyway, thanks for watching, and have a good rest of your day.